What if I told you that one of humanity's oldest enemies has learned to hack your immune system like a master spy? The tuberculosis bacterium doesn't just hide from your body's defenses, it actively sabotages them, turning your own cellular security system against you. And scientists have just discovered exactly how it pulls off this molecular heist. This isn't just biology, this is biological warfare at the cellular level. Welcome back to Mind and Marvel, where we explore the most incredible scientific discoveries that reveal the hidden battles happening inside our bodies. Today, we're uncovering one of the most sophisticated immune evasion strategies ever discovered. Tuberculosis has been humanity's companion for millennia. Even today it kills 1.5 million people annually and infects about 1/4 of the world's population. What makes TB so successful isn't just its ability to spread, it's its remarkable talent for living inside our immune cells while staying completely invisible. Think about that for a moment. Your immune system has specialized cells called macrophages, literally big eaters, whose job is to consume and destroy invading bacteria. But TB doesn't just survive inside these cellular destroyers. It thrives there for decades. Scientists have just published groundbreaking research in Cell reports that reveals exactly how TB pulls off this incredible feat. Mycobacterium tuberculosis evades host immune defenses by using its protein kinase PknG to disrupt the linear ubiquitin chain assembly complex, LUBAC, impairing NLRP3 inflammasome assembly. What they've discovered is that TB bacteria are essentially molecular hackers, using a special protein weapon called PknG to systematically disable your immune system's alarm system. It's like a burglar who not only breaks into your house but also cuts the phone lines and disables the security system. So how does a microscopic bacterium outsmart one of the most sophisticated immune systems in the animal kingdom? What makes this molecular sabotage so effective? And what does this discovery mean for developing new treatments for TB? Let's dive into the fascinating world of bacterial espionage. To understand how TB hacks your immune system, we first need to meet one of your body's most important security guards, a molecular complex called the NLRP3 inflammasome. This isn't just any immune component, it's like the fire alarm system of your cells. The NLRP3 inflammasome is a multi-protein complex involved in the release of mature interleukin, 1b, and triggering of pyroptosis. Think of it as a sophisticated security system that can detect when dangerous invaders are present inside your cells. When your cells detect signs of infection or damage, the NLRP3 inflammasome springs into action. It's like a smoke detector that doesn't just sound an alarm. It also calls the fire department, activates the sprinkler system, and starts evacuating the building. Here's how it works. When the inflammasome detects danger, it triggers the release of powerful inflammatory signals called interleukin, 1b, and interleukin 18. These molecules are like emergency broadcasts that alert the entire immune system to the presence of invaders. But the inflammasome has an even more dramatic response called pyroptosis. Essentially, it triggers the infected cell to sacrifice itself to prevent the infection from spreading. It's like a building's self-destruct system that destroys the structure to prevent a fire from spreading to neighboring buildings. This inflammatory cell death serves multiple purposes. It eliminates the infected cell, releases inflammatory signals to attract immune responders, and exposes the invading bacteria to the harsh extracellular environment where they're more vulnerable to immune attack. Against most bacterial infections, this system works beautifully. The inflammasome detects the invaders, sounds the alarm, calls in reinforcements, and eliminates the threat through controlled cellular suicide. It's a brutal but effective defense strategy. The inflammasome itself is a marvel of cellular engineering. It requires precise assembly of multiple protein components, proper cellular signals, and careful regulation to avoid accidental activation that could damage healthy tissue. One crucial component in this process is something called LUBAC, the linear ubiquitin chain assembly complex. LUBAC acts like a molecular construction crew, helping to properly assemble and activate the inflammasome when it's needed. Under normal circumstances, from the moment a cell detects danger to the point where it triggers pyroptosis and releases inflammatory signals, the entire process takes just minutes to hours. It's a rapid response system designed to contain threats before they can spread. But what happens when this sophisticated security system is sabotaged from the inside? What occurs when the invaders figure out how to disable the alarms, jam the communication systems, and prevent the self-destruct sequence? That's exactly what TB has learned to do. Now let's meet TB's secret weapon, a protein called PknG that acts like a molecular saboteur, systematically dismantling your cell's security systems from the inside. PknG phosphorylates the Lubac subunit Hoyle-1L to prevent it from properly functioning. PknG is what scientists call a protein kinase, essentially a molecular tool that can modify other proteins by adding phosphate groups to them. Think of it like a molecular hacker that can reprogram other proteins by changing their chemical signatures. Here's the brilliant part of TB's strategy. Instead of trying to hide from the inflammasome, PknG directly targets Lubac, the complex responsible for properly assembling the inflammasome. It's like disabling the construction crew before they can build the alarm system. Specifically, PknG phosphorylates a protein called Hoyle-1L, which is a crucial component of Lubac. When Hoyle-1L gets phosphorylated by PknG, it can no longer do its job of helping assemble the inflammasome properly. Imagine Lubac as a team of construction workers trying to build a fire alarm system. PknG is like a saboteur who drugs the workers' coffee. They're still physically present, but they can't perform their essential tasks. The alarm system never gets built, and the fire goes undetected. When Lubac can't function properly, the entire inflammasome assembly process falls apart. The cell loses its ability to detect the TB infection, can't trigger the inflammatory response, and fails to sacrifice itself to contain the bacteria. The result is devastating for the immune system, but perfect for TB. Instead of a rapid, coordinated immune response that would eliminate the bacteria, the infected cell becomes a safe haven where TB can multiply and establish a persistent infection. But PknG doesn't stop there. The protein also triggers NLRP3 degradation, actively destroying the inflammasome sensor, even if some components manage to assemble despite the Lubac sabotage. It's like the saboteur not only prevents the alarm system from being built, but also destroys any alarm components that manage to get assembled. This dual-action approach ensures that the cell's defenses are completely neutralized. The sophistication of this strategy is remarkable. PknG doesn't just randomly damage cellular components, it specifically targets the exact mechanisms that would be most effective at eliminating TB bacteria. It's precision sabotage at the molecular level. Other bacteria have evolved various ways to avoid immune responses, but TB's approach is uniquely sophisticated. Instead of just hiding or running away, it actively rewires the host cell's security systems to create a safe operating environment. Laboratory studies show that when PknG is active, TB bacteria can survive and multiply inside macrophages for weeks or months, turning what should be their executioners into their protectors and homes. Here's where TB's strategy becomes truly diabolical. Not only does it disable your cellular alarms, but it actually uses your immune system's own cells as luxury apartments, living inside the very cells meant to destroy it. When a macrophage encounters TB bacteria, it does exactly what it's supposed to do. It engulfs the invader in a process called phagocytosis. The macrophage essentially eats the bacteria, trapping them inside specialized compartments called phagosomes. Normally this would be the end of the story. The phagosome would fuse with lysosomes, cellular garbage disposals filled with powerful acids and enzymes that break down and destroy the captured bacteria. It's like a cellular death chamber. But TB has learned to prevent this deadly fusion. While the PknG protein is disabling the inflammasome alarm system, other TB proteins are sabotaging the phagosome-lysosome fusion process. The bacteria effectively jam the locks on their cellular prison. The result is that TB bacteria end up living inside specialized compartments within macrophages, protected from the external environment but not subjected to the usual deadly processing that would normally kill them. Inside these sabotaged phagosomes, TB bacteria don't just survive, they thrive. They multiply slowly but steadily, turning each infected macrophage into a bacterial factory that's invisible to the immune system. The infected macrophages often form structures called granulomas, organized clusters of immune cells that surround the infection. While this might seem like an immune response, it actually serves TB's purposes by creating a protected environment for long-term survival. This leads to what's called latent TB infection, a state where bacteria are present and alive in the body, but not actively causing disease symptoms. The person feels healthy, but they're carrying potentially millions of TB bacteria in their lungs. Remarkably, about 1/4 of the world's population has latent TB infection. These people aren't sick, aren't contagious, but they harbor TB bacteria that could reactivate if their immune system becomes compromised. If the host's immune system is weakened by age, illness, stress, or other factors, the dormant TB bacteria can reactivate, multiply rapidly, and cause active tuberculosis disease. It's like having a sleeper cell of bacteria waiting for the right moment to strike. What's particularly clever about TB's strategy is that macrophages are relatively long-lived immune cells. By turning them into safe houses, TB ensures its own long-term survival without having to constantly find new cellular hosts. Recent research suggests that TB bacteria inside macrophages can even communicate with bacteria in other infected cells, coordinating their behavior and responses to environmental changes. It's like a hidden bacterial network operating inside the immune system. Understanding exactly how TB sabotages the immune system isn't just fascinating science, it's opening up entirely new strategies for treating one of the world's most persistent infectious diseases. Traditional TB treatment requires taking multiple antibiotics for 6 to 9 months, and drug-resistant strains are becoming increasingly common worldwide. The fact that TB can hide inside immune cells and disable cellular defenses explains why these infections are so difficult to cure. Many current antibiotics struggle to penetrate inside macrophages where TB bacteria are hiding, and even when they do, the disabled immune responses mean the host's immune system can't effectively help clear the infection. But the discovery of PknG's sabotage mechanism suggests a revolutionary new approach. What if we could disable TB's sabotage tools instead of just trying to kill the bacteria directly? Scientists are now working on developing specific inhibitors of the PknG protein. These drugs wouldn't necessarily kill TB bacteria, but they would restore the infected cell's ability to detect and respond to the infection. If we can prevent PknG from sabotaging Lubac, and the Inflammasome infected macrophages would regain their ability to trigger inflammatory responses and undergo pyroptosis, exposing the hidden bacteria to immune attack. Even more promising is the potential for combination approaches, using PknG inhibitors to restore immune function while simultaneously treating with traditional antibiotics to kill the now exposed bacteria. This represents a shift toward what scientists call host-directed therapy. Instead of just attacking the pathogen, we're enhancing the host's natural ability to fight the infection. It's like repairing the sabotaged security system instead of just sending in more guards. The discovery also has huge implications for TB vaccine development. Understanding how TB evades immune responses helps scientists design vaccines that can trigger the right types of immunity to overcome TB's sabotage strategies. There's even potential for personalized approaches, testing individual patients to see how effectively their immune systems can detect TB, and tailoring treatments based on their specific immune response patterns. Researchers are also exploring whether existing drugs approved for other conditions might be able to interfere with TB's sabotage mechanisms, potentially speeding the development of new treatments. Of course, developing new TB treatments is a long process requiring extensive testing for safety and effectiveness, but understanding the molecular mechanisms gives researchers much more specific targets to aim for. If successful, treatments that restore immune function could be particularly valuable in areas with high rates of drug-resistant TB, where current antibiotics are failing and new approaches are desperately needed. The TB discovery is part of a larger revolution in understanding how pathogens manipulate immune systems. TB isn't the only organism that has learned to hack our cellular security. It's just one of the most sophisticated. Many viruses, bacteria, and parasites have evolved ways to interfere with inflammasome function, suggesting that disabling cellular alarm systems is a common strategy in the microbial world. Remarkably, cancer cells often use similar strategies to avoid immune detection, hijacking many of the same pathways that TB manipulates. Understanding bacterial immune evasion could inform cancer immunotherapy development. The research also sheds light on autoimmune diseases, where the immune system attacks healthy tissue. Some autoimmune conditions might involve dysregulation of the same inflammasome pathways that TB targets. Even normal aging involves changes in inflammasome function, and understanding these pathways could lead to interventions that help maintain immune function throughout life. This research opens doors to understanding how we might deliberately modulate immune responses for therapeutic benefit, enhancing immunity when fighting infections, but dampening it when treating autoimmune conditions. So there you have it. The tuberculosis bacterium has evolved into a master molecular spy using its PknG protein weapon to systematically disable your immune system's alarm systems and create safe havens for infection. We've learned how TB targets the Lubac complex to prevent inflammasome assembly, how it triggers degradation of immune sensors, and how this allows bacteria to live undetected inside the very cells meant to destroy them. While TB's immune evasion strategies are remarkably sophisticated, understanding these mechanisms at the molecular level gives us unprecedented opportunities to develop new treatments that restore immune function and help the body fight back. This research represents hope for the 1.5 million people who die from TB each year and the billions more at risk of infection. Scientists around the world are building on these discoveries to develop the next generation of TB treatments. If discoveries like this that reveal the hidden molecular battles inside our bodies fascinate you as much as they fascinate me, make sure to subscribe to Mind and Marvel and hit that notification bell. We're living in an incredible time for understanding the microscopic wars that determine our health. I want to hear from you in the comments. What aspect of TB's immune sabotage surprised you most? Can you think of other examples where understanding disease mechanisms has led to breakthrough treatments? What questions do you have about immune system function and bacterial infections? Remember, every time scientists uncover the molecular secrets of pathogens like TB, we get one step closer to winning the ancient war between humans and infectious disease. The battle is fought at the cellular level, but the victories benefit all of humanity. Thanks for joining me on this incredible journey into molecular espionage and immune system sabotage. Until next time, keep marveling at the hidden battles that shape our health and survival.